Hey, so whenever I first made this PowerPoint, I made it using PowerPoint, and I didn't realize whenever I uploaded it into a Google slide that I would lose the sound. So here's a video of me going through some of these slides with you, and just hopefully hearing my voice and explaining it might clear up some confusion. So use this video as needed. So this first slide talks about diabetes through time and how we've treated it differently in the early 1900s to current treatment. And this unit starts off with diabetes and how we treat diabetes. But the big question at the bottom is, where do we get insulin for patients? So this is the question that's going to tie into our current lab. Insulin is a protein that our body naturally makes, unless you're like type 1 diabetic, or sometimes if you have insulin resistance, you have to make a certain type of insulin in order for it to stimulate the body to accept it. And so we get insulin from bacteria. We actually insert a gene that is the code for making insulin, and we put that gene into bacteria. The bacteria will read the gene, express it, and it will make insulin. In this slide, it's explaining how insulin is made in a more detailed step in a process called recombinant DNA. We actually learned about recombinant DNA and how it works in Unit 1 whenever we learned about making vaccines. If you remember, we had a paper plasmid. You had to choose the correct enzyme to cut your plasmid with, and then you inserted that new piece of gene into the plasmid in the Unit 1. In this one, it's showing that you first start off with a vector. In this case is a plasmid. You will cut the vector using restriction enzyme, and you will grab DNA from whatever species that you're interested in getting the gene from. Then you use ligase to paste this gene of interest into the vector plasmid. Once this vector plasmid has the new gene, the plasmid can be forced into bacteria via the process of transformation. And now this bacteria that has the plasmid will read the plasmid and express the genes on the plasmid, including our target gene that's been added. So in this case, we would just essentially add the target gene of insulin, and once the bacteria has that gene, it will make and express insulin. So we will not be making insulin in the lab or in the classroom. Instead, they have us making what's known as Gfp. So of course, as always with all these slides, you'll want to pause me and read through this. But Gfp is a fluorescent protein that glows in the dark. So different than insulin, obviously, we're going to be able to physically see whether or not the protein was made because it glows in the dark. You could, or with UV light. So you should be able to see it. The connection between insulin, how we started off this PowerPoint, and Gfp is it's the same process. Just as we talked about in the previous slide where you have to put the gene of interest for insulin, you would have to put the gene of interest for the Gfp gene, and essentially transform bacteria to take in the plasmid that has the gene of interest for the Gfp gene, and the bacteria will make it. So same process, just different product. Instead of making insulin, we're making Gfp because it's a easier, more visible protein to do in the lab. This slide is very important. You will want to draw and write all this information in your lab journal. So to start off, you have the origin, and this is mentioned and allows for the DNA sequence, this plasmid, to be copied and made in the bacteria. Then we have the T7 promoter and the Gfp gene. The T7 promoter recruits a particular type of RNA polymerase that is needed for the Gfp gene to be expressed and turned on. So essentially the T7 promoter is an on or off switch that we will talk about a little bit more later. The GFP gene is the gene that is the instructions for making the GFP protein. So it's very important in our lab, as all these really are. And then we have the AMP resistance gene. The AMP resistance gene makes the bacteria resistant to ampicillin, an antibiotic, whenever it has this gene. So a big question I like to cover is, why do we have an AMP resistance gene on the plasmid? Why would we want to make bacteria resistant to antibiotic ampicillin? Isn't that dangerous? Don't we try to prevent bacteria from becoming antibiotic resistant? So we're going to talk about it a little bit more on the next slide. The AMP resistance gene acts as a selectable marker. And so what that means is, if you have a plate of LB agar, which LB is literally a broth, it's just bacteria food, and you had some bacteria that took in this plasmid, and you had some bacteria that did not take in the plasmid. It does not matter on a regular LB agar bacteria food plate whether or not it has a plasmid. All the bacteria will grow on it. So you should have a mixture of some bacteria that has a plasmid and that will glow because it has a gene, and some bacteria that do not have the plasmid and will not glow, which is okay, but we tend to want to eliminate the ones that do not have the plasmid. We want all the bacteria that only have the plasmid. They're the one that's interesting. So in order to get only bacteria that have the plasmid, all you have to do is make a bacteria plate that has LB agar, but also has ampicillin in it. And that way, if you plate any bacteria on this plate, it would have to have this plasmid and would have to have the ampicillin resistance gene in order to grow on the plate. And so if there's any bacteria that did not get the plasmid, it will die because ampicillin will kill it. And so essentially on a plate that has ampicillin in it, you would only have the bacteria that took in this plasmid, and so all the bacteria should glow for you. It acts as mentioned, a selectable marker to ensure only the bacteria that has the plasmid will grow on a plate with ampicillin. Let's see. So in order to control when the bacteria will make the protein GFP and when it will glow, scientists have added what's known as an inducible promoter. That's an important term. An inducible promoter acts as an on or off switch for certain genes. So in this case, the T7 promoter is acting as an inducible promoter that is able to turn on and off the GFP gene, as we will talk about more on the next slide. So, normally, we're going to ignore some of this right here, but you have this lac promoter, and what a lac promoter does is it creates this little red half kind of circle known as a lac repressor. And in normal conditions, the lac repressor will actually attach to this T7 polymerase gene, and it prevents this gene from being read. The purpose of this gene normally is to make T7 RNA polymerase. This is that particular type of polymerase we talked about in previous slides that plays a part in expressing the GFP gene and turning it on and reading the gene. So you want to have the T7 RNA polymerase available to read the gene in order for the GFP protein to be made. So as mentioned, this lac promoter normally makes these little red half circles that attach to the T7 polymerase gene, and it prevents it from making the T7 RNA polymerase. So if the T7 RNA polymerase is not made, then the green fluorescent protein is also not made because it cannot be read. So in order to essentially turn on this gene with the lac promoter, all you have to do is add a substance called Iptg. And what Iptg is, it's like a chemical, and if you just simply put it in the bacteria plate, Iptg will come in and it will bind to these lac repressor like enzymes. And essentially whenever the Iptg binds to these enzymes, it keeps the enzymes busy and they can no longer attach to the RNA polymerase gene. They essentially have their active site taken, and so they cannot attach here because it's busy with Iptg. So whenever the Iptg is present, the T7 RNA polymerase is turned on. It makes T7 RNA polymerase, and T7 RNA polymerase can then go and read the Gfp gene and make the Gfp protein. So in big terms, because this can be a lot, the Iptg is an inducer and is needed for the expression of the T7 RNA polymerase. You have to have Iptg in order to make T7 RNA polymerase. T7 RNA polymerase is needed for the expression of GFP. If you do not have Iptg, then you will not make the GFP protein, because that gene cannot be read. So for this video, I have 15 minutes on each video. I'm at 12 minutes now. So I'm going to end this first video at slide number 9. And then in part 2, I will start up on slide number 10 to finish up this slideshow presentation.